So we have CRISPR, now we know how to deliver that CRISPR inside of the cell. So once we deliver that, we can do many things. We can do many kind of, we can apply for many purposes. So for therapeutics, it can be used, gene and cell therapies, antimicrobials, animal health. We can do basic biology research. We can do agriculture biotechnology by doing precision breeding. We can bring novel trait. We can influence crop genetics by these. And also industrial biotechnology like microbial fermentation, therapeutic bioproduction, etc. I will briefly mention some of the big applications which are already happening. So this slide shows you how CRISPR application happens in human and for disease discovery, disease research, or gene therapy. So you can see cancer therapy, different cancer models could be made, animal models could be made by CRISPR-Cas9, cancer immunotherapy, genome edited immuno T cell can be made. And in human trial, already one such system in human trial in China, esophageal cancer PD-1 gene modified to resist cancer. So in fertility related research, in vitro gametogenesis, gene knockout to stop miscarriage. In Francis Crick Institute, UK, people are using CRISPR-Cas9 to stop miscarriage of embryonic cells. Different genetic disease such as cystic fibrosis, genetic blindness, Huntington's disease, beta thalassemia, etc. People are targeting and trying to modify or do therapy by CRISPR-Cas9. Similarly, infectious disease can also be targeted in similar way. So University of Pittsburgh is using CRISPR-Cas9 to shut down HIV replication. Lot of work happening in Kobe University as well. And also organ transplantation. We know that different organs, animal organs are, people are trying to use for human, like liver or whatever. So these organ development could be expedited or modified as per our need by using CRISPR-Cas9. Also in cancer, different kind of cancer models. I'm not going in detail. This will be available later on. One can see it. Different areas of cancer, this CRISPR-Cas9 can be used. And also drug discovery, CRISPR-Cas9 plays a big role, starting from target identification, compound screening, hit validation, lead identification, and finally clinical trials. So any compound you found, you develop animal model by CRISPR to test those compound, or even it's a natural compound, then you can change the production process of that compound by using CRISPR-Cas9. So a lot of work is happening in this area, and almost more than 1,000 Cas9 based such gene drug discovery activities are under pipeline for the regulators. So it's happening very rapidly. Also diagnosis, you know, this is our paper in preprint, which will come out in a quite renowned journal very soon, where we basically accumulated different kind of CRISPR-Cas9 based targeting of the COVID-19 disease, SARS-CoV-2. So CRISPR-Cas9 can be used by much faster in a point of care way for identification of different nucleic acid detection based disease detection. So COVID is one of them. Similarly, CRISPR-Cas9 people are using for animal model system and disease research. So these monkeys, rhesus and cyanomegalous monkeys, resembles humans in evolution, anatomy, physiology, and pathology. So they are widely used. So this picture is from one of the research outcome where BAML1 gene is knocked out. So circadian rhythm, our timing sense is destroyed in these monkeys. So sometimes they sleep when in the daytime, or they are awake in the night time. So their circadian rhythm is changed, and people are doing many kind of research to find out how that works and what kind of drug could basically help in insomnia and other kind of diseases. Similarly, muscle-related things, these Beagle cloned and done by CRISPR-Cas9 for extra muscle growth, and different research is going on by using CRISPR for animal model system. Similarly, for pet breeding, like Dalmatians are very well-known and popular breed. They have certain kind of genetic mutation that makes them prone to suffer from bladder stone. By CRISPR, people have shown that these bladder stone related gene could be shut down, and similarly, faster horse breed can be made, healthier fish can be made, and in dairy, we know there are all these cows, they're having these horns and they fight with each other. So all dairy system farmers, they cut those horns, and very crude and brute way of handling them. But by changing a single gene, you can do the dehorning. And there are a lot of examples. People have made dehorned cows, so they don't need to dehorn physically. And so it can be used greatly in animal and livestock. Similarly, what we are working at Reliance, we basically work in the industrial biotech area and select crop biotech area. So there are a lot of possibilities are there, starting from yeast, algae, in vitro enzyme production, plant cell mediated chemical production, filamentous fungi mediated chemical production. So all these bioproduction system in industrial fermentation, basically what happens, cells manufacture a product. So now detergent enzymes, ethanol, pharmaceutical drugs, vaccine can be produced inside of the cell. And CRISPR system can be used to make production cells better factories. So this is another area, industrial biotechnology, a big area we are working. CRISPR can be used in this area for terpenoids, alcohol, amino acid, antibiotics, lipids, anti-tumor agents, polyhydroxyalkanoids, phytochemicals, organic acids, many such thing development. We can use CRISPR from different kind of cell and cellular organisms. So CRISPR-I and CRISPR is greatly used nowadays in bacterial metabolic engineering as well for certain kind of product development. And as I mentioned, algae is greatly used. So we use algae, this is our farm, and we use CRISPR and other gene editing and other tool to change microalgae so that we can make bulk chemicals, biofuel, lipids, biogas, or pigments. So for different chemicals from algae and obviously biofuel by applying CRISPR. So far what I have shown is basically CRISPR mediated different thing development, different important chemical development, and CRISPR, what is CRISPR? Its basics, from where it was discovered, what kind of modifications, what kind of application it could be done. And recently, there are many modifications have been made in CRISPR, which has wide application in animal, human, industrial biotech, as well as crop biotechnology or agriculture. So I will run through some of these modifications. So we know that if you take a Cas9 and hook one cytidine deaminase in the Cas9 and you send that target, what happens? It can change a single base. So single nucleotide polymorphism can be changed by attaching one deaminase molecule with the Cas9, which is nothing but we call as base editing. So base editing, there are three types of base editing can be done nowadays: C to T conversion, A to G conversion, or A to I conversion in a RNA. So different kind of further modification has been done to make it more precise. I'm not going in detail. All the references and some of the major things are here. Later on, you guys can have a look. And also, people have developed CRISPR-X, which is nothing but they have added MS2 hairpin with gRNA. So MS2 hairpin can hold many deaminase. So what happens? Wherever you target that, it goes there and can do many changes. So within a certain area, you can do targeted point mutation. Instead of one, you can do many by CRISPR-X. Also, people have used CRISPy. It's a new technology called Retron. So people use Retron. What is Retron? Retrons are natural DNA elements coding for a reverse transcriptase as well as a template on which the RT acts to create a multi-copy single-stranded DNA. So how this was used? So retron is fused with gRNA. So now you have a Cas9 and gRNA and then you attach a retron, which is basically a template and a RT enzyme. Now the template, you can design that template, you can introduce your gene of interest there, and then this whole thing can go inside of the cell. Template can be made in front of the cut site, and homologous recombination can happen, and that template can get inside of the cell by this CRISPR technology, a modified CRISPR technology. Similarly, people have used Evolve-R. People have developed Evolve-R. What is that? So Evolve-R consists of an error-prone polymerase. So this one error-prone polymerase, you fuse with a Nuclease, nicking Cas9, which is nothing but cuts only one strand instead of two strands. So once the Cas9 cuts the DNA, it gets dissociated. Now error-prone Paul basically attaches there and try to rectify, and it gives rise certain kind of mutations. Now you can use many gRNA and target this whole system inside of the cell in many places of the genome. So your Cas9 will go and cut, Paul will rectify and bring different mutations. So you can generate a pool of mutants within very short period of time for your research. So this is called Evolve-R. So you can change in a tunable window, you can change randomly by point mutation, creating many point mutations. Similarly, people are using for gene knock-in, in case of plant, we know we use agrobacterium for plant transformation. And that agrobacterium has many Vir genes. So now certain Vir genes can attach to the right border. Now you take a gene of interest, attach a right border sequence with it. Then with a Cas9, you attach a Vir gene. So that Vir gene will bind with this sequence supplied by you, and then gRNA will take that whole thing in a certain area of genome. And in the cut site, you will get that gene of interest in larger copy, and that can be introduced inside of the cell by homologous recombination. So Vir-D2 mediated homologous recombination can also be done. Recently, people have found that certain kind of transposes, jumping gene elements, can also do gene insertion. So this is Feng Zhan's lab from MIT. They found that certain kind of cyanobacteria, they are having this kind of CRISPR-associated transposes, which basically helps in gene integration. So a lot of happenings are going on, lot of additions, new development. And the last one, which I would like to mention, is prime editing. This is a new technology, came out recently. People used in animal and human cell line, and now this has been used in plant as well. It's a great technology because instead of gRNA, you take a PEG RNA. It is a different kind of targeting. You take a PEG RNA and your Cas9 protein, attach it and with a reverse transcriptase. Now PEG RNA will supply your gene of interest, and reverse transcriptase will copy that. And by CRISPR-Cas9 activity, that PEG RNA containing DNA will be copied inside of your target area by reverse transcriptase. So this is a great tool people are using. So you don't need to do any break. You don't need to do any double stranded break. So off target or other kind of things are not there. It hybridizes in 3 places, not only gRNA, it hybridizes in 3 places. So off target is not that prone, not off target prone. No need of repair template co-delivery. So you are sending everything together, and precise point mutation and small indels can be done by prime editing. So it's most advanced modified version of genome editing, which can be applied for gene insertion greatly. And in plant, it can be used for many purposes, like small segment insertion, small segment deletion, introduction of multiple point mutation, introduction of replacement of desired amino acid, correction of undesired mutation, introducing premature stop codon, introducing single point mutation, etc. So with this, this is my last slide. A new happening are coming out. So artificial intelligence people are using for CRISPR application. Now, as I mentioned, you take your gRNA, you take your Cas9 protein, you send inside of the cell, it will cut and do the change. Now, how this change will come up? Because after cutting, cell will try to repair, there will be mutations. Now, there are ways. Previously, people thought that mutations are random. But nowadays, scientists have found that these mutations are not random. Cell also has an algorithm to basically change these or bring these mutations. So in Delphi or Sprout or Forecast in silico tool software has been developed by different universities and institutes where you can basically predict this change, and based on the prediction, you can manipulate your experiment. So it's great AI tools are coming up every day. Recently, there is a one-seq tool which will come up very soon from University of Harvard, which I have seen in one of the recent symposiums, where very precisely you can also predict if there is any off-target, which is very high throughput system. So this is where I finish my first part, where I introduced basic CRISPR, its applications, its recent modification discoveries for applying in animal and human. So 45 minutes I have taken already. So I may need another 15 to 20 minutes to finish the second part. Might be a little more, but if time is not there, I can squeeze a little bit. So now the thing is, in agriculture, how this CRISPR system is used and what are the greatness of this system, why it is used. Previously, what happened? You send a tDNA with your gene of interest, and tDNA stays inside. So your foreign element stays inside to make a GMO. But for CRISPR-Cas9, you send a tDNA get inserted in the genome. Then the tDNA will give rise a CRISPR-Cas9 tool. The CRISPR-Cas9 tool will do the change, this red color area. And then in the next generation, the tDNA will be segregated out. So you will discard that progeny. You will keep that progeny which has only change. So what is there? What we have got here, no foreign element is there, no insertion element is there. You just make change in certain bases inside of the genome, and you accept that clean element or clean progeny or clean plant for your scientific purposes or application purposes. So this is the basic change CRISPR has bring, no foreign element, only your changes. So why this is so important? It has another one applications, a great applications. So if we do breeding in a traditional way, then we take our germplasm of interest, then we take important donor variety, we cross them, we get a progeny, then we do back crossing of that progeny for 5 to 7 generations, and then it takes 8 to 10 years to develop a new product or a new crop which can be used for certain purpose. So there are mutational breeding process as well nowadays. Worldwide, people are using it for many decades, and by using chemical or radiation, that also take 8 to 10 years. Transgenic breeding, if you want to develop GMO, that also quite time taking because the regulatory aspect is pretty long, and it takes 8 to 12 years to make GMOs and bring in market. And you know there are a lot of issues with social acceptance, etc. But for genome editing, without insertion any foreign element or without keeping any foreign element inside of the genome, you can do it very fast within a year or two. And even if you use downstream integration breeding, you can do total thing within 4 to 6 years. And we see that regulation is much, much less hurdle for genome edited product in different part of the world, especially in US, in Canada, in Latin American countries, in Japan, and many other countries. In Australia, people are considering, the regulators and scientific boards and bodies are considering this simple genome editing, which resembles natural mutation as natural product. So you don't need to go for lengthy regulatory process and cost. So there are mainly 3 types of editing can be done in agriculture by this genome editing. First is SDN1, where you send the genome editing tool, you cut, cell repairs, so you get a mismatch of small indel. The breeding equivalent, the native breeding equivalent, if we see it, is just by a mutational breeding. It's just type of mutational breeding. In rice, many trait has been developed by mutation breeding, like RC, FRG1, WAXI, XA13, male sterility, etc. So, which has been done by mutational breeding. You can do it within in 1 year by CRISPR. So this is for agriculture. The second type, SDN2 type, where you insert an ortholog via homologous recombination. So this blue part is your gene area which you want to change due to certain reason. So you give a 2 cut at 2 end of that region, and then you send your gene of interest, either developed from either taken from a similar plant genome, or you develop it, whatever it may be, or take from some other organism, and you insert it by homologous recombination, which is known as allele replacement or allele swap. And this is basically a similar equivalent of breeding technology called MABC, which is marker assisted backcross breeding, backcross hybridization. We develop near isogenic line by this process, this breeding process, which takes, again, 8 to 10 years to develop. That can be done in 1 generation, in 1 year, and then downstream applications. So SUB1, PUP1, Saltol, AG1, XA genes, PI genes, these are very important characters people have brought inside of the rice system by using this marker-assisted backcross breeding, by working 8 to 10 years for each of these gene applications, but which can be done by CRISPR in 1 shot. Now, the third type of genome editing which can be done is SDN3 type, which is basically, there is no breeding equivalent of it. It is just similar to GNA development, GMO development. But you can take a whole gene, you insert it by CRISPR-Cas9, and you get a GMO type of element, which is similar to GMO, but little bit more cleaner. You don't need to have left border, right border, all these things, less foreign elements, just your gene of interest by homology-directed repair. So Roundup or BT resistance kind of characters, which people have or industries has developed as a GMO, can be developed by SDN3 as well. So 3 types of genome editing can be used in plant: SDN1, SDN2, SDN3. So now, if you see this equation, this is basically breeder's equation for genetic gain, breeder's equation for genetic gain. So whatever way you do breeding, or whatever way you do genome editing, or whatever backcrossing, or you use this formula of genetic gain. Now forget everything, just think about breeding cycle and cost, L and C. So if you can reduce breeding cycle by twice, your genetic gain increases by twice. So CRISPR can do that. CRISPR can bring down your breeding cycle greatly, your breeding cycle, and that will increase your genetic gain. Also, it can bring down your cost significantly. CRISPR can also influence your genetic standard, selection accuracy, selection intensity, etc. I'm not going in that detail. I would concentrate here that for commercial breeding purpose, end of the day, you have to make seed, you have to bring that in market. Either you do it by public sector, university, institutes, government, or you do by industry. Whatever you do, end of the day, time and money matters. So CRISPR-Cas9 can greatly do good in that area with respect to our present breeding activities. So there is limitation. What is that limitation? So CRISPR in this area, this subtle area, CRISPR-Cas9 can work greatly. But at the bottom area, CRISPR-Cas9 still cannot be used. Why? So the complex traits, poorly characterized trait, CRISPR-Cas9 cannot be used. None of the system cannot be used. Trial and error breeding integration can be done and checked, but you need to understand the biology greatly, you need to understand the pathways greatly, and you need to understand gene-to-gene relations greatly to find out which gene to change and which gene to modify. This is less of a problem for genes like height determining gene, well-characterized disease gene, biochemical grain quality gene, physical grain quality gene, submergence, salinity, etc. These are all either single gene or few gene pathway which are well known. We can apply CRISPR and immediately change it. But for complex traits like yield, like drought, etc., where cascade of genes are involved, it is still a challenge. So we need to know biology greatly to apply CRISPR-Cas9 properly in agriculture. Already there are many CRISPR-Cas9